Good day. In today's lecture, we will be discussing about how to study gene expression. In particular, we will be looking into the microarray and the RNA-seq data analysis. Basically, what are the things to consider when we are using the data from the RNA-seq and the microarray methods. So for a start, we have several researches here. I got this from Nature about DNA gene expression data. So we have actually a lot of publications and studies that are studying here. It's actually one of the hot topics because, for example, in the field of drug design, it is one of knowing the gene expression analysis is also one of the ways to identify targets for drugs, as well as for the diagnostics. We can determine, or we can use biomarker genes in order to help in the diagnosis of the early onset of some diseases, specifically the cancer. So transcriptomics is the study of the transcriptome. So what is the transcriptome? The transcriptome is the complete set of the RNA transcripts that are produced by the genome under specific circumstances or in a specific cell. So even though all of the cells in our body have the same set of genes and have exactly the same DNA, basically our cells are clones of each other, but the way they express those genes are different. So basically, for example, our skin cells express a different set of genes as compared to the cells in our brain. And the same goes for the cells in our bone marrow. They express a completely different set of genes as compared to the cells in, for example, our white blood cells. Even the white blood cells, depending on the type, we have the B cells, the T cells, helper T cells, natural killer cells. So all of them, even though they are white blood cells, all of them express different set of genes. So they have different transcriptomes. And this expression of the transcriptome also varies according to time. So in fact, even when, for example, the same set of B cells, same two B cells, if in the presence of a pathogen or the antigen, the B cells will express a different set of genes as compared to when it was not exposed or when there is no antigen present. So you have different expression depending on the time, the conditions, and the type of cell. So we will have a lot of information to get that. So we are using high throughput methods such as microarray analysis, to actually understand these transcriptomes. So comparison of the transcriptomes allow the identification of genes that are differentially expressed in distinct cell population. So in doing one of the ways to study the transcriptome is through comparison. So we have, what we will peg as a normal condition, or one condition, this is a sleeping or inactive condition, for instance, then the active condition. So we usually have comparisons. And that is where the beauty here comes from. So in studying the transcriptome, we can learn a lot by actually crafting our questions carefully. So how, for example, we want to know the activity in the B cells. What genes will be active for an active B cell. So our comparison will become a B cell in an inactive state and a B cell in an active state. So that could be one way of comparison, or even a naive B cell and a B cell that has been exposed to your pathogen. So depending on exactly what you want to know, we can craft many different types of study design here. So the key here one of the key here in this research is knowing or crafting a very good study design. Okay, so in one of the ways to actually determine one of the high throughput ways, when we say high throughput, these are methodologies that provides us with a lot of information. So one of those high throughput ways is what we know as the microarray. So in a microarray, you have your normal cell and you have your cancer cell. So you have two cells that are being compared. So these two cells can be a normal or a diseased cell. This cell can be, for example, one cell on one part of the body versus another cell on another part of the body, or it can be a cell in a different state, active or inactive. So depending on your study design, on your research design, you will choose the two types of cells that you will be comparing. So you will culture those one cell, you will clone them, so you will produce cultures of those. This is to amplify the signal. And then you will isolate the mRNA. So from the mRNA, you will create a cDNA. cDNA is the complement DNA. It's a DNA obtained from reverse transcripting your mRNA using the reverse transcriptase enzyme the from a viral reverse transcriptase enzyme. Example of a virus with a reverse transcriptase is actually HIV. So it's actually a useful tool. That is reverse transcriptase. So you will tag them with two different colored probes. So the usual color are green and red. So these are complementary colors. So it's more distinct when you view them. And then you hybridize the cDNA probes to oligosequences on a microarray. So the microarray is basically a chip. The size of the chip is just the size of a small microscope slide. So that's just about the size of that. So in this chip are embedded the genes. So you have the known genes, or the genes that are found, or basically we have different microarray sets or chipsets depending on what type of condition you are looking for. For example, breast cancer, Parkinson's disease. So each of these chipsets contains the embedded genes that basically your chipset have these genes arranged in an array. So when you have an array, it's basically rows, and this here is an array. So you have rows and columns. So we know, or rather, based on the chipset, we know the identity of the genes in this cell here is known. This one is also known. This one is also known. So basically, what happens here is your cDNA from the normal and disease cells, they will be mixed and hybridized to the chipsets. So when you hybridize them, it will find their complement. So basically, it will bind to the complementary sequence. So you will see here, after hybridization, the fluorescent probes will light up the colors of your gene. So here we have we can know, or we can then compare which gene is over or underexpressed. For example, this one here is gene 1. So we know the identity of this gene. So here it's a gray color, so there is no fluorescent probe here. That means that neither the cancer cell here and the normal cell here are present or hybridized to this one. So there is not enough transcriptome or gene expression from both normal and cancer cells. So for a yellow one, so yellow color comes from the all co-equal combination of yellows and reds. So this is basically light mixing, not the painter's color mix. It's the mixing of light. So when you mix the green and the red light, you get a yellow light. So anyway, so here you have there is no significant difference basically between the cancer and the normal cell. So basically, they are present on both in equal measures. And then you will have cells or genes that lights up green or red. So when you have green, so that means that the dominant color here is the green, which comes from the normal cell. So here, if you compare it, for example, this is the normal condition, this is the diseased condition. So that means that this type of gene is underexpressed in the diseased condition because it doesn't show up with the diseased cell, it shows up in the normal. And then for a red, so that means the dominating gene here, or the dominant cDNA here, comes from the cancer cell because it's the one that's labeled with red. So that means that this gene is, the red ones, is overexpressed in your cancer cell because there's a lot that comes from the cancer cell but not enough from the normal cell. So basically, these are basically one of the ways to compare. Now, there are many different possibilities here because there might be a yellowish green color. So does that mean is it significantly present or not significantly present, or rather significantly underexpressed or not significantly underexpressed? So basically, what comes here is knowing the exact frequency of the colors that you will get, and then calculating its statistical significance. So basically, the key, one of the important things in the studies of transcriptome is statistical significance. So we have in the NCBI a tool used to analyze microarray data. So you have the Geo2R, which is an interactive web tool that allows the user to compare two or more groups of samples in a geo series in order to identify genes that are differentially expressed across experimental conditions. So basically, Geo2R is a tool that is used in studying microarray data because when you perform or basically the data that are obtained from microarray are usually submitted in the gene ontology database in NCBI. So this is so because the data here is high throughput. Even if you're the one who actually did the microarray studies, it's very difficult to actually harvest everything on that data. So sometimes some other scientist or researcher might be able to make use of the data for his or her own research design or study, or even to compare on his or her own data. So basically, they put it in the repository so that other scientists can also refer to that data that they had, and it might also help in their research. So the Geo2R is basically a tool to help other researchers to analyze, or even your own the researcher himself who made that data, to analyze these microarray data that have been submitted. So the results are presented as table of genes ordered by its significance. So which is significantly overexpressed or significantly underexpressed. So here is the direct link to your website. And then we also have the gene ontology omnibus. So I'm talking about the geo dataset. So geo means the gene ontology omnibus. So it's an international public repository that archives and freely distributes microarray, next generation sequencing, and other forms of high throughput functional genomics data submitted by the research community. Basically, here we can see the communal, or basically the community-wide research teamwork that have been doing. So that actually is present. It's one of the hallmarks we have of this research that we have right now. So in doing research, unlike in the old days, centuries ago, or even a century ago, research is now accessible anywhere across the globe. So a researcher in the US can help, or can the data from the researcher in the US can be accessed by a researcher in Asia, or even in South America, or even in Europe. So you have a lot of basically going around here. So likewise and vice versa. So data from researchers in Asia and the Pacific, and then it can be accessed by people, researchers in the US or in Canada, or even in Europe. So basically, you have here a community of researchers working together in a global scale. And this is actually made possible by the use of this public internet database that you can access actually anywhere, basically with the use of the internet. So questions microarray data analysis seeks to answer. So basically, as we demonstrated, we can compare two conditions. So what are those two conditions? Example can be the deceased versus the normal condition of the cell. We can actually also see cell differentiation. So for example, what is the difference between a helper T cell and a cytotoxic T cell? And then it can also be for different physiological conditions. So under stress, are the genes that we express, and when we are relaxed, these are the genes that we express. Or for example, even for the brain cells relaxed versus brain cells stressed. So we can also do comparisons across multiple conditions, and we can do data clustering as a function of the sample, or as a function of your genes. What are the clusters of genes that are involved, etc., etc. So we actually, depending on your research question, and how to craft your study design, you can microarray can be a powerful tool. So here I will also demonstrate, or show to you. Actually, we are running out of time. So in the next, this will be in the next part of the video. So we will look at RNA-seq analysis. So this is a different approach to microarray, but also in transcriptomic data.